Hi, my name is Maria Munoz. I'm a second-year PhD student at UC Davis, working under Dr. Pamela Line. In the lab, our research is on acquired epilepsy after acute intoxication to the organophosphate DFP. Research has shown that acute intoxication to DFP can lead to neuroinflammation, neurodegeneration, acquired epilepsy, and cognitive impairment. A lot of studies have been done in the adult brain, but there have been fewer in the juvenile brain. These studies have also shown that among the brain regions damaged are the dentate gyrus and the piriform cortex. So in this study, we will be looking at those two regions. In the juvenile brain 3 months after acute DFP intoxication, and we will be looking at two outcomes. One is neurodegeneration, or the degeneration of neurons, and neuronal hyperexcitability detected by CFOS immunoreactivity. So in this first figure, we are looking at the number of degenerating neurons using a chemical stain called FluoroJC in the dentate gyrus and in the piriform cortex. DFP is our group exposed to the organophosphate. Naive and vehicle are our control groups. And we found that there was no increase in neurodegeneration at 3 months after acute DFP exposure. In the next slide, we are looking at CFOS immunoreactivity. We're using CFOS as a potential marker of neuronal excitability, which may indicate acquired epilepsy. We're looking at this in the dentate gyrus and the piriform cortex across those 3 different groups. Naive vehicle, which are our controls, and DFP, our experimental group. Visually, we did not see a difference. And then we quantified it in 2 different ways, looking at CFOS density, which is the number of cells per millimeter squared of tissue, and also the percent of CFOS positive cells. And again, we found that there was no significant difference between our experimental group relative to our control in either brain region. So in conclusion, our preliminary findings find that there are no significant increases in neurodegeneration in either brain region, and there was no significant increase in CFOS immunoreactivity or neuronal hyperactivity either. But future studies are needed. So other studies will look into neuroinflammation, which is another outcome of acquired epilepsy. So I would like to thank the ARC Foundation for providing the funds to do this research.